Yeah, it's fantastic to be here and part of this series. I watch lots of these online, and I hope that this will make a interesting contribution to some people at least. So my title today is a little bit vague. It's about chromosomal rearrangements and adaptation, and I want to emphasize some of the sort of confusing and unexpected findings that we've had in a few years of looking into this topic in this butterfly, Danaus chrysippus, that I'll introduce in a bit. Just to say that I am from the University of Edinburgh, where I am part of the Institute of Ecology and Evolution there, and I have a small group, and I'll be presenting work from about the last 5 or 6 years from some of the postdocs and PhD students that I've supervised. So I was going to start over here with a slide that I think is very familiar to us from reading lots of papers coming out nowadays, where people look for patterns associated with local adaptation between species, in this case FST, allele frequency differences between populations. And we see these striking patterns with very, very low differentiation across much of the genome, and then these big blocks of differentiation that, in this case, correspond with peaks, with known or suspected chromosomal inversions. And this has now become a very familiar sight, and not really an unexpected sight, because there's quite good theory to suggest that when we have populations adapting to different environments with gene flow between them, there are selective forces that can favor recombination suppression, which is what chromosomal inversions do. So just a very quick summary of some of the earlier theory on this. If we have more than one locus under selection for local adaptation, here for 2 environments, then we have immigrants that come in and their alleles are selected against, but these immigrants also cause a problem for locals that carry local alleles, because when they mate with immigrants and recombination occurs, then we get recombination load. So we get these local alleles that are co-occurring with unfit foreign or incoming alleles. And so if we were to have a mechanism to suppress recombination between local alleles, then this, on average, will increase the fitness of the carrier because it avoids that recombination load. And so that can, under certain selection parameter, model parameters, I guess, select for that reduced recombination mechanism, say a chromosomal inversion, to spread in the population, especially if you consider the fact that it is linked to the alleles. In this case, it's carrying the alleles that are locally adapted. This model has been shown to work under certain circumstances. It requires that there's gene flow happening, because that is what drives the selection that favors linking these locally adapted alleles. But the parameter range may be quite narrow under this model. What you may remember, if you were at Katie Lotterhaus's seminar just over a month ago, is that there are models of polygenic adaptation that expand on this. And I've actually stolen, well, with permission, some screenshots from Katie's talk. You might remember this idea that under local adaptation with polygenic evolution, where new mutations can arise during the simulation, then an inversion may capture some locally adaptive alleles and start to spread. But its spread can be helped along by the fact that it can acquire additional locally adapted alleles. And these are all at small effect loci that contribute to a polygenic trait. And that can increase the likelihood that inversions are involved in local adaptation. So I now think it's quite intuitive that under gene flow, when we have traits that are contributed to by multiple loci, that we may expect to see things like inversions involved. This is another slide that comes out of one of the simulations from Sarah Schall, working with Katie and Ben Haller, where they showed that these inversions increase in their effect size through time during the experiment. And that's because of the accumulation of these additional variants over time. And something that's relevant for today's talk is you also see this compounding of inversions on the same genomic loci as they capture new combinations of alleles in the same parts of the genome. So we may expect that these inversion patterns will be present all over the place, and we don't necessarily see that, and so we have to think about what types of mechanisms might select against them or might impede their spread. And there has been some theoretical work on this. One of the things to bear in mind is that when an inversion is rare in the population, it tends to be found in the heterozygous state. And if it really does suppress recombination in heterozygotes, then a rare inversion is going to have very few opportunities to recombine. And so there's a period when it's at low frequency in the population where it suffers from Muller's ratchet, the inability to purge deleterious mutations. And this could, in fact, cause an inversion to become, go from being something that increases fitness to being something that decreases fitness. It may be selected out of the population, or it may eventually be held in a kind of, as a polymorphism in the population, potentially with heterozygote advantage over dominance due to a kind of masking effect where these recessive deleterious mutations are masked in the heterozygous state. And this was suggested in a recent review paper by, a model paper by Paul J, which I actually haven't cited here, as a mechanism that could cause something that is locally adapted to never actually fix in the local population because it picks up these deleterious alleles along the way. So this is just something to think about when we go looking in the wild for why things might not neatly match our expectations. Another idea that was put forward by Marius Rösti a couple of years ago is the idea that, well, if your environment changes, then this locking together of locally adapted alleles that we saw here might not actually be a good thing in the long run, because at some point, say we've got a new environment here on the right, where the optimal genotype is now a mixture of alleles from the first two environments, it might be impossible to create that genotype once we've fixed for an inversion that locks together these fit alleles from the second environment. My analogy for this is your chocolate cake and your vanilla cake. You can have a chocolate cake or you can have a vanilla cake, or you can recombine them, but in this case you cannot have your cake and recombine it. And so, yeah, there may be some costs to these mechanisms. So just a reminder about why chromosomal inversions suppress recombination. There are two main mechanisms by which this happens. The inversion can pair up correctly in meiosis. I'm going to get there with the pointer, and form a loop like this. And when we have a crossover in that loop, we get these unbalanced gametes forming where the products of meiosis loop back on each other. And I've only shown the recombinant products here, but 2 of the 4 products of meiosis would, in this case, probably lead to inviable offspring because they carry duplicated and missing parts of each chromosome. Or we could just have this mechanism that I've called shifted crossovers, where the crossovers don't happen within the inverged part of the genome, but tend to happen outside of it. And that could happen if there's just failure to correctly pair during meiosis. But recombination, we know, is not completely suppressed within rearranged regions, at least inversions. There are 2 mechanisms by which it can occur. The one is if we get 2 crossovers happening within this loop, a double crossover event, that leads to exchange of material without any imbalanced gametes being formed. And the other mechanism is non-crossover gene conversion, where a double-stranded break occurs and a crossover event is initiated but doesn't complete, and a small amount of material is copied from one of the 2 haplotypes into the other. So these are mechanisms by which some mixing can still occur within these parts of the genome. And they may act to nullify some of those potentially costly mechanisms that I was talking about before. And so I think we still need to do a lot more studies on how these parts of the genome behave in the wild to understand how they persist and which of these mechanisms sort of dominate the mechanisms such as accumulation of deleterious mutations or can those typically be overcome by enough recombination? And one way to do this is to just do deep dives into certain test cases, case studies, and that's what I'll be talking about today. This species, Danaus chrysippus, is the African monarch, also called the African queen or the plain tiger, and it's well known in Central and Eastern Africa for being very polymorphic. And it was once thought to be a sort of system under balancing selection, until it was later realized that this part of Africa where you get all this polymorphism is just a sort of multi-way hybrid zone between otherwise distinct populations. This is a milkweed feeding species like the monarch, and it is aposomatic. It's recognized by predators as being distasteful and avoided. But it has these different morphs that tend to have different geographic regions. I mentioned the hybrid zone, so that's this region over here where all 4 of these morphs showing on this slide co-occur, as well as intermediates between them. But then there are regions such as Southern Africa and West Africa that are mainly monomorphic. Southern Europe and North Africa is a part of the range where the butterfly is expanding, and we see some interesting polymorphism there between this lighter colored morph that you find in Asia and the darker colored one that we find in Southern Africa. This map was actually made from citizen science data. This is quite an abundant species, and this is just a good example of how records from people just taking photos of butterflies can be used to learn some interesting things. And we actually use this data to think about how these traits are distributed across space. First thing I wanted to say about this is these are all genetic traits. In fact, they were shown to be genetic in crosses back in the 70s with a simple genetic basis. There's thought to be a single locus that controls whether you have white on the hind wing, whether you have a dark background or a light background, and whether you have a black tip on your forewing with a white band or not. And these last 2 loci that control melanism and the forewing tip are thought to be tightly linked, and that was shown in crosses. So when we break this down into traits controlled by what we think is a single locus, we see that the allele for white hindwing is found only in West Africa, and through space it sort of follows a very broad cline over hundreds of kilometers to being in very low frequency in East Africa. And similar for this allele that gives you an absence of the forewing tip, if we go in space from East Africa down to Southern Africa, we see again a very broad cline. And so what we imagine is happening here is that this is a very dispersive species, and selection is favoring local wing patterns. So just to go back here, and we think this selection is imposed by predators that recognize the local most recognized warning pattern and select against warning patterns that they haven't learned to avoid. And that once these patterns become established as the common local pattern, there's positive frequency dependent selection maintaining the local warning pattern. So we obviously want to understand the genetics of this, and we've gone out sequencing, and we've sampled with the help of lots of collaborators who I'll list at the end, from many different sites across the range, and each of those morphs that I was talking about, as well as in the hybrid zone. And next, I will show you that FST plot that everyone shows. So this is what ours looks like. This is comparing pairs of populations from different parts of the range, and it got that familiar pattern, extremely low FST. In fact, the main point I'd like to make here is how low FST is between populations that are sampled thousands of kilometers apart. So this isn't like what we see in many taxa where we can detect isolation by distance on the scale of tens or hundreds of kilometers. Here, if I had to give you, say, chromosome 10 or 11 from one of these individuals, you wouldn't be able to say where in Africa it was sampled, at least not with much success. But then there are these chromosomes with very clear peaks of differentiation between the populations. And we've mainly focused on this one on chromosome 15, because it differentiates all pairs of populations we look at. And we assumed very early on that this must be controlling wing color differences because it's pretty much the only difference between this East African and Southern African population. I'll come back to the other two peaks right at the end if I have time. So we, from our population sampling, have done a genome-wide association study. This is a Mendelian trait, so even from fairly meager sampling, we're at about 200 individuals now, although this was based on a smaller subset of that. You can very easily pick up that chromosome 15 is indeed the only locus that really strongly associates with this presence-absence of the four-wing tip, whereas the brown-orange hindwing, or wing background color at least, is associated with chromosome 15 as well as some other points along the chromosome. There's a really, along the genome. There's a really nice peak here on chromosome 15, which I'll zoom in on in a second. In fact, here it is. So at the top there is that sharp peak at the one point on chromosome 15 for the background color phenotype. And at that locus is the gene yellow, which is a part of the melanin synthesis pathway in insects. So that makes sense. Now, if you were thinking about recombination suppression in inversions, and now looking at this GWAS plot, you might think, well, how is it that they are able to get such a narrow peak of association in a region of the genome that is supposed to be not recombining between morphs? If you are thinking that, that's the right question to ask, and there is an answer to that question coming in a few slides. The other locus, we're not really able to narrow down its association. It's somewhere in a part of the chromosome that you can see I've actually chopped out. So I've left out about 7.5 megabases of the chromosome, and that's because there's a big copy number variable part that's extremely difficult to genotype. And it's quite possible that this C locus is actually inside that bit of the genome. And I'll explain a bit more about that copy number variable part in a bit. The thing that intrigued me most when I first looked at these results, though, was the fact that we see an FST plot that has a peak between all three morphs. So how does an inversion cause differentiation between three different groups in pairwise directions, since there are only two arrangements of an inversion, a forward and a reverse? So clearly this can't be a single inversion if it's suppressing recombination between more than two things. So we suspected that we'd have a complex arrangement. And yeah, here we go. How do we explain this three-way recombination suppression? So to answer this question, my two postdocs at the time, Kang-Wook Kim and Rishita Kain, took on the challenge by just simply assembling chromosome scale genomes for each of the morphs. And fortunately, in this butterfly in particular, it's quite easy to assemble whole chromosome contigs from long-read data without needing any scaffolding. So we managed chromosomal assemblies, and this colorful part of the chromosome is the part of the chromosome at which, when we map reads from known morphs, we see elevated divergence from the incorrect or the non-matching morph. So that should be the part of the chromosome that is differentiated between morphs. But the other thing you might notice is that these assembled chromosomes are very different lengths. And that was the first clue that there are some significant structural differences between them. So I'll just summarize the structural differences between them with this next slide. So this is showing bits that are collinear in gray or bits that are inverted in red. And we can see that there are multiple inversions that differentiate these morphs. But then there are also these huge gaps where I've not really aligned anything. And these correspond to these many green fragments here. And each of these light green fragments shows a fragment of an ancient single copy region, which was duplicated over and over again in the evolution of this lineage. So we can show that by comparison with an outgroup. It's not shown on this slide. But this is a region that expanded from about 1 megabase to over 5 megabases through repeated duplications. We're not really sure about the mechanism nor the selective pressure that drove that. But you can see how having different copy numbers of a copy number variable region like this would also contribute to suppressing recombination, because chromosome pairing would really be impaired. So there's a number of different mechanisms that contribute to the low recombination that we see between the morphs. And we also know that these accumulated over time. And we can learn a bit about this by looking at the tree. And this is a summary of what we've learned. It's quite difficult to show you all the results because we made trees for different parts of the chromosome. But the summary is that we think that the copy number variable region expanded first. And that's because there's members of the genus quite far diverged that already have high copy number in this part of the chromosome. And then after that, translocations and inversions of different regions occurred. We know that inversions can occur through a mechanism of non-allelic homologous recombination. So when copies of repeated parts of the genome pair up incorrectly and recombine, then inversion can occur. And so it's likely that once this region had expanded in copy number, it made sort of fertile ground for subsequent inversions to occur. And that's how we've ended up with the morphs that we have today. And we know that these inversions occurred deep in time because species that occur in different continents carry some of them. So we think they occurred pre some of the speciation events that led to Danaus chrysippus existing as it is today. So how are the haplotypes evolving now? That's really what I want to talk about for the bulk of the rest of the talk. We wanted to understand, not just what is their structure, but what's going on at the sequence level, and how much variation is there in these populations. So one of the first things we thought we would do is sequence more individuals and see if we really see them clustering into the 6 possible genotypes that we should expect to occur in the wild. 3 types of homozygotes and 3 types of heterozygotes. So you might recall from other talks seeing, and papers of course, seeing PCAs for inversions where you see 3 clusters corresponding to the 2 homozygous genotypes and the heterozygote. In the case of a 3-way rearrangement like this, we should see 6 clusters in a PCA. And we almost see that. We do see the 3 clusters that should correspond to homozygotes, here at the bottom and the top right and the bottom, the top left. And then maybe 3 clusters that could correspond to heterozygotes. Although things start getting a bit messy, there are sort of individuals that don't cluster anywhere, and maybe too many clusters in some places. So we started to think, well, perhaps there's more going on than we thought based on wing patterns alone. We wondered whether there are other haplotypes, other arrangements out there that we hadn't assembled, or maybe recombination going on at some level, producing some of these intermediate points. The other way we thought to look at this is with a phylogenetic network approach. So this is what a distance-based neighbor net network looks like for this part of the chromosome. And again, we can make out the clusters that we know to correspond to homozygotes of each morph. And we know that from the resequencing data that they tend to be homozygous. But then there are individuals all over the tree that we can't really explain as simple intermediates that you'd expect from heterozygotes, except maybe this group up here that seems to be intermediate between these 2. And so we thought, well, why don't we, rather than assuming that there are 3 progenitors of all this variation, why don't we try to do a naive clustering using a tool like structure or admixture and ask it how many groups it thinks there are, how many non-recombining haplotype groups are there out there. So we're modeling these as if they're kind of populations where the recombination between them is, you can think of as gene flow between populations, if indeed there is recombination between them. And when we did that, we actually got a result out that suggests that there are probably 6 diverged clusters. So we had only described 3, and there are probably another 3 more out there that we didn't know about because they are phenotypically cryptic. So this is our sort of best understanding now. I've only shown 4 here as homozygous because there are only 4 that occur in the homozygous state. So this is quite a lot going on. I hope you can see that on the tree, I've now got little circles, little pie charts. So the pie charts show whether the genotype of an individual is assigned 100% to a certain cluster or 50%, as we'd expect if it's heterozygous. So we have homozygotes down here, up here, and over there. And then we have homozygotes for this pink cluster, just a couple of them occurring in a group over here. So there are at least 4 divergent groups that are not recombining. And these plots of chromosomes are ancestry painting. So this is a method sliding along the chromosome and assigning individuals into each group according to its SNP variation, using a small number of individuals from each group as reference individuals. Obviously, we use the homozygotes as reference individuals. But then there are 2 more colors on this plot that are quite hard to find. One of them is up here, where it's a sort of purple color, and we can tell that this is a haplotype that must be very closely related to this dark red one, because some of the red-purple heterozygotes are here very, very close to this homozygous red group. We actually know what this is. We know about another rearrangement that's out there. And in this case, it's really confusing because it's a fusion to the W chromosome that occurred just a few thousand years ago. And it was this haplotype here that fused to the W chromosome. Females don't recombine in Lepidoptera, so if you fuse to the W chromosome, you stop recombining. So that seems to be another way that this chromosome has stopped recombining. But because that particular haplotype is only ever present in females, and it's only ever present on the fused to the W chromosome, it's never present in the homozygous state. So we were unable to use that for ancestry painting. So we don't have any individuals that are painted with that ancestry. And then the 6th haplotype group is here on the left. And these are a group of individuals that appeared to be painted as homozygous for this 6th group. But when we looked into it, we found that they're actually a type of heterozygotes. So we've got all the possible kinds of heterozygotes distributed around the plot here now, heterozygous for each of the colors that are out there, except for these ones here that we expected to be homozygous. According to our ancestry painting, they're actually carrying 1 haplotype that matches this red one and 1 haplotype that is something that we were unable to paint with any color because we don't have any homozygotes of that morph to paint with. And when we did try to assign this as homozygous and paint with it, we got a meaningless result. So at the time, we hadn't sequenced any homozygotes. We've now, quite recently in 2024, we actually did manage to find some homozygotes of this morph. So maybe one day I'll come back and show that there are more different kinds of homozygotes and paint with another color here. But clearly there's more going on here than what we thought. And I still haven't explained some of the points on this plot, such as this one down here that seems to be intermediate between all the other groups, and these 2 over here. I mentioned before that there may be recombination contributing to this, and indeed, that's what we see. So I'll just leave this slide up for a bit. These are a sampling of some of the recombinant individuals that we see that we can't explain with any single haplotype. Sometimes the recombination events are very clean and beautiful and actually occurring between rather than within inversions, because remember there are multiple inversions in this rearrangement. And a recombination event that occurs between 2 adjacent inversions shouldn't cause unbalanced gametes. So these type should be quite common, and indeed that seems to be a very common point of recombination that we see over and again in this supergene. But then there are also ones that look like this one on the top left, or the far left here, where there's evidence of recombination within an inversion, giving this mosaic pattern here. So we definitely think that there is recombination occurring within the inversions on some level. And you might have noticed that that pattern of a mosaic recombination actually shows up in several individuals in exactly the same pattern. So that looks like a recombinant haplotype that formed once and has increased in frequency. Thinking about the geography again, things tend to make sense. We tend to be homozygous for a single haplotype with a single ancestry in each of the monomorphic populations. In the hybrid zone, this is sampling in Rwanda, we get all of the different haplotypes as well as recombinants between them. Not surprising, they will have the most opportunities for recombination there because that's where the most heterozygotes are going to be occurring. And then the one cryptic result was this Southeast African population where we get this pink haplotype that we weren't aware of. The phenotype it's associated with is indistinguishable to our eyes from that in the rest of Southern Africa. And so this was one of the more than meets the eye results that perhaps, and it was a big surprise as a butterfly person, perhaps color pattern isn't the only thing under selection in this species. Big surprise. Maybe there are other traits that matter for ecology, and maybe, in fact, this pink haplotype here is adapted to some other environmental parameters than just what the birds are able to see. So the question is, have we looked at UV patterns? We have not. We have all the wings, though, of all these sequenced individuals, so that is something we could do. Birds can see UV, yes. I mean, it certainly seems possible, although I guess there's no reason. What I didn't say is there are about 150 genes in this part of the genome. So there's no reason there shouldn't be other traits beyond wing pattern involved. But yes, that is one of the things that we have not yet got to. I've added two more populations now on the slide. One is down here on the bottom left. This is a population from a small island, St. Helena, which has a weirdly high number of recombinant individuals. And we think this population is quite recently colonized and very strongly bottlenecked. In fact, we've shown that all 6 of these individuals are highly inbred. And so the persistence of polymorphism and recombinant haplotypes is quite strange. We think it might have to do with relaxed selection on that population. And then here a population from Southern Europe carrying 3 copies of that cryptic haplotype that we weren't able to assign to a group. Well, that we have a little more information on now, but we were quite surprised to see polymorphism. This is the range margin here, and these populations are thought to also be bottlenecked, and so quite interesting that we're seeing polymorphism in 2 of the sort of recently colonized marginal populations. So we wanted to understand the origin of these new haplotypes. Now I'll go back a slide quickly. Particularly this one in Southern Africa here. Oh, I've lost my pointer, there it is. Which is associated with an indistinguishable phenotype. When we plotted differentiation along the genome, or genetic divergence rather, we found that it was actually most closely related to the East African one, which we call the Klugii haplotype, even though its phenotype is most similar to South Africa. So I suspected that perhaps the origin of this haplotype involved recombination. So to look at this, we've used topology weighting, which is a method that can assign or at least weight parts of the chromosome according to how much they support each of several possible subtree topologies. And so you can imagine the whole big complex tree of all these individuals, and we're sampling little clusters of 3 tips at a time, and then asking how closely related is the Klugii individual to Karamu, that's the pink Southern African, and to Orientis, that's the blue Southern African one. And we see that throughout this mixed part of the chromosome outside of the supergene, there is no signal. That makes sense. They're completely freely mixing there. But within the supergene region, we see that there is strong support for this pink haplotype grouping with the East African one rather than Southern African throughout most of the supergene, in fact. And it's only when we look more closely, that plot at the top is a smoothed plot. I'm now zooming in on this one inversion over here that's shown in purple, and showing the raw results of this topology weighting. We can see that it is really a mosaic where Karamu, the southeastern African haplotype, is sort of sometimes East African and sometimes Southern African in its ancestry. And the part that I was most interested in is on the far left here, where there's a box, because that's where we have the gene yellow. And if we zoom in on that, and now this is ancestry painting again with a tool called LOTA, which has a hidden Markov model, which finds the best breakpoints between the ancestries. These two lines in the middle here are two individuals that are homozygous for the pink haplotype. But if we force them to be painted either as yellow or blue, they are clearly mosaic ancestry. So this really looks like the product of recombination that has neatly placed this block over here over the gene yellow into the yellow genetic background. A bit confusing that blue has been placed into a yellow background at the gene yellow, but there you have it. And this is the locus where we in fact saw that strong association for controlling wing color pattern. And this is probably why we're able to see such a strong association, because at some parts of the supergene, recombination has broken down these co-associations that would usually just obscure a genome-wide association study identifying the specific locus that controls a trait in a region of suppressed recombination. We found another anomaly in North Africa where there were individuals that had the red haplotype group, but they also had dark wings, which we didn't expect. And so we also did exactly the same thing. We took those dark individuals and we asked, how do they cluster? And they mostly do cluster as red individuals. But again, here at the yellow locus, we found that they have a slice of blue haplotype. And indeed, they carry a slice of blue ancestry. And this time it really carves out just the promoter region of the gene yellow. So once again, this is a completely independent event where a dark haplotype, just for dark coloration, has recombined into a different genetic background, this time in Southern Europe. So these plots are quite complicated, but I've got a summary slide over here. This is our starting point where we had these three haplotypes we had assembled. We've now discovered there's a fourth one in Southeast Africa, which we believe arose through recombination that put this wing pattern into this background. I didn't talk about the C locus because that's the one in that copy number variable region, but we assume that that has also recombined into this background to give us this wing pattern. And so, yeah, as I said, our best guess is that there's some combination of selection pressures here in Southeast Africa that favor a recombinant haplotype. So this was exactly the kind of problem that Maria Swersti paper was suggesting supergenes or inversions would have, and perhaps they don't have that problem if there's enough recombination happening within them to create these new combinations. And then in Southern Europe and North Africa, we have a dark haplotype that again seems to have arisen through an independent acquisition of these dark coloration alleles. And you can ask, well, why didn't the Southern African entire haplotype just invade there if selection was favoring a dark phenotype? And it might be that this red haplotype contains alleles at other loci that for some reason are favored in this northern part of the range. And then there was that other mystery haplotype that I mentioned that also occurs in North Africa and Southern Europe. We didn't even know what traits it affected or what outcomes of it would have on the trait because it was heterozygous whenever we found it. We have gone back and sampled some homozygous individuals now, and we can say that it is associated with the light wing pattern. And so now there are two ways of making a light wing pattern coexisting in this marginal population at the very edge of the range. And this is a mystery to us. We still do not know why this would be occurring. This seems to be under balancing selection because when we looked at the demographics of this population, we see it's very recently or very strongly declined. On Africa, this species has a huge effective population size of around 5 million, but here this was from a population in Southern Europe. It is really in just the low thousands, and the individuals have large blocks of homozygosity, so it's inbred, and yet it persists as polymorphic with these 3 haplotypes really, and 2 ways of having the same light color morph. So there might be some balancing selection going on here. And one suggestion is that perhaps one of these haplotypes was spreading, but picked up those deleterious alleles I spoke about early on and has been unable to fix. These are 2 master's students that I had last year in the lab, Chongyang Wang and Benyat Yanez. And Benyat asked about deleterious variation by quantifying the ratio of non-synonymous to synonymous polymorphism, which is a measure of the efficacy of selection in the population, and finds some tantalizing evidence, perhaps, that he's used different colors here, but in yellow are homozygotes for this mystery haplotype, and they seem to have some excess of deleterious variation relative to the other haplotype. This is what we'd expect. This is the rarer of the haplotypes. It occurs less often in homozygotes and gets less opportunities for recombination, so may have less efficient purifying selection. But why this would maintain it is unclear unless there's some kind of heterozygote advantage, perhaps through this associative overdominance mechanism, where there's masking of deleterious alleles on both haplotypes in the heterozygotes. We did try to look for this in the form of a deviation from Hardy-Weinberg equilibrium, but our sample size was around 100 individuals, and we didn't detect any statistical excess of heterozygotes. But that's not to say it's not there just at a selection strength that we can't detect with such a small sample size. So I just wanted to end with a final story, going back to these other 2 peaks here on chromosomes 4 and 7 that I didn't talk about. This is more of a warning that, again, things may be more complicated than originally suspected. I think as a butterfly person, I often come at the problem initially thinking about wing colors and not really thinking about other traits. In this case, these 2 peaks seem to differentiate the West African population from everything else. And so we suspected that one of these peaks presumably is what gives the West African population its white hindwing. We did suspect that they were both inversions, and in that experiment where we assembled haploid genomes to look at chromosome 15, fortunately one of these inversions was segregating, and so we were able to show indeed that at least the one on chromosome 7, oh wait, no, this is chromosome 4. That arrow is pointing in the wrong chromosome. Anyway, that one is an inversion. The chromosome 7 one probably is too, but we haven't assembled it yet. So which of these 2 inversions carries the white hindwing locus that we see in West Africa? My postdoc, Bruna Kammer, in the lab was doing lots of breeding for various experiments, and so we took some of her broods and she mapped the white hindwing trait, and the answer is that neither chromosome 4 nor 7 is controlling the white hindwing of West Africa. In fact, chromosome 8 shows the strongest association, and she's repeated this with another cross now and shown again that it's chromosome 8. So just a warning to not assume that you know the trait under selection. The odd thing is that there is no FST peak on chromosome 8 differentiating West Africa from the other populations, not even a hint, in fact, of an FST peak. So where is this white locus that seems to be strongly selected in West Africa and absent everywhere else? We don't know. What we do know is when Bruna tried to narrow down her association using a GWAS of a wild population rather than QTL mapping in a brood, her association also went away. So we can find the locus segregating in a cross like a Mendelian locus, but when we look in a wild recombined population, there are no SNPs that associate with this trait anywhere. And I'd be interested to hear anyone's guess. Our best guess as to what's happening here is that this trait is controlled by a structural variant, probably a copy number variant, in a part of the chromosome where we've been unable to genotype anything, and that the recombination rate is so high that the drop-off of association is so rapid around this functional site that even within probably a few kilobases where we are able to genotype SNPs again, we're not seeing any association there. I don't have any real evidence to support this yet. We need to assemble a genome from both morphs, I think, to look at the structural variation in chromosome 8 better. What I can say is the region around okay, so the question is how inbred are the two initial genotypes? In this particular result I'm showing here, these were wild broods from wild parents. We've also repeated it with an inbred line. So we've seen this association on chromosome 8 in 3 or 4 different families. We haven't done that here. Right, absolutely. There could be shared ancestry giving us this peak. I think the fact that we've seen it in multiple different crosses from multiple different sets of parents always going on to chromosome 8 doesn't make that a bit less likely. But yeah, this is a fairly early stage result, so I agree that we do need to rule out confounding factors. But even so, if it's not chromosome 8, then where is this Mendelian locus? Our best association on chromosome 7 does happen to be, sorry, chromosome 8, does happen to be in a region of reasonably high recombination rate compared to some other parts of the chromosome. So it could be that recombination is indeed obscuring the signal, but there still needs to be a functional locus somewhere there. And that's kind of all I have to say about this quest to find that other color patterning locus that sometimes it's not in any of the inversions. And I'll end there just to conclude by saying that, yeah, we went looking in the wild population, and I think it was very rewarding. So I'd encourage others studying rearrangements to not just assume that they know which variants are out there, and rather go sequencing if you can, because we found a surprising allelic diversity, including cryptic haplotypes that associated with what to us were indistinguishable phenotypes. And we think this is partly fueled by that acquisition of additional structural variants. We still don't know for these new haplotypes we've described what structural variants are suppressing recombination in those, but presumably there are additional inversions and translocations out there. And then, of course, recombination has been part of the fuel, which maybe overcomes some of the limitations of suppressing recombination in the first place, the fact that some recombination is still happening. But there are probably other selective drivers out there, not just color pattern. And it's probably likely that once a rearrangement becomes established locally in a population, there's no reason why other traits may kind of jump on the bandwagon, or other selective drivers may cause the accumulation of additional variants associated with other environmental adaptations to accumulate in the same part of the genome. Unfortunately, we're still quite ignorant as to what those may be. And finally, we find something that we cannot explain, which is unexpected polymorphism in these bottlenecked range edge populations that we still don't fully understand. Whether this is preserved polymorphism because of associative overdominance, or just a lack of purifying selection, or some kind of balancing selection. I think some other kind of balancing selection. Those are all possible explanations that we hope to solve in the future. And I will stop there. Happy to take questions, and thank you very much for listening. And of course, thank you to a very large number of people, especially all the fieldwork helpers who I haven't mentioned so far, who contributed to the sampling and breeding in this work. Thank you.